Welcome back to the Deep Dive, where we take complex fields of research and distill them down to the essential, mind-blowing facts you need to know.
Today we're moving far beyond the first stages of embryogenesis.
We're getting into the lifelong developmental processes that really define our adult existence.
Yeah, when you say development, people immediately think of the embryo, right?
Of course.
But the architecture of the adult organism, I mean, how we determine and maintain our final size and incredible diverse mechanics of repairing missing parts, that is a continuous, fascinating act of development. It never truly ends.
That's such a great way to put it. The embryo is maybe chapter 1 of the story where the blueprint is laid down.
Absolutely. And today we're diving into, let's say, chapters 19 and 20. This is all about the continuous engineering and, the maintenance of that blueprint.
So we're exploring how an organism can scale up its body parts, sometimes by a billionfold, from a single cell to an adult, and keep it all proportional.
And then, crucially, how it maintains the capacity to repair or even rebuild complex anatomical structures throughout its entire life.
So our mission today is to unpack the fundamental rules governing these processes. We'll be looking at the master molecular switches.
The signaling pathways that act as the ultimate architects of size. Think of them as a metabolic accelerator pedal on one hand and a kind of physical boundary fence on the other.
And then we're going deep into the wild diversity of cellular strategies that organisms use to regenerate.
From, true pluripotent stem cells in flatworms to specialized cells that literally undifferentiate themselves in a newt's limb. It's incredible stuff.
Okay, let's start with the big question of scale. When you look at any adult animal, a dog, a whale, a mouse, their final size is incredibly consistent within their species.
It is.
Which suggests there's some kind of hard stop, a predetermined boundary. How does the body know exactly when to stop growing?
That is one of the most fundamental questions in developmental biology. And the answer is, well, the body isn't just growing until it hits a certain age.
It's not just running out of steam.
No, it's actively working toward a specific, finite, genetically determined size. If you look at the classic growth curve for most animals, it's characterized by incredibly rapid growth at first.
Almost exponential.
It can look that way, yeah. But then the curve decelerates really smoothly and tails off until the growth rate hits zero. And it hits zero precisely when that species-specific adult size is achieved. It's a beautifully regulated slowdown.
But we do see a few famous exceptions to that finite growth, don't we?
We do. Some animals, particularly certain types of fish, are often described as having indeterminate growth.
Right.
But even here, that's a little misleading. They might have a theoretical maximum size, but their growth slows down so drastically in late life that they might never actually reach that maximum within a normal lifespan.
I see. So for the vast majority of mammals and insects, the target size is finite and pretty much non-negotiable.
Exactly.
Okay, so if we're talking about growth, what is actually expanding? Is the body just making its cells bigger, or is it making more of them?
That's a key distinction. For the vast majority of tissues, and for general body growth, it is overwhelmingly an increase in cell number.
Okay, so more cells, not bigger cells.
Right. Cell size itself is a remarkably conserved property across different species. Cell division is typically triggered when a cell reaches a specific threshold size.
So if the cell doesn't grow enough, it just won't divide.
It won't. And if it grows past that threshold, it initiates mitosis. So cell size is essentially a checkpoint for division, not really a strategy for increasing organ size on its own.
But there are exceptions.
Of course. You have to acknowledge the specialized exceptions, which are primarily found in what we call postmitotic tissues.
Tissues where cells stop dividing early on.
Precisely. Think of large parenchymal cells like hepatocytes in the liver or cardiomyocytes in the heart. These cells can increase their mass and function without dividing.
How do they do that?
By increasing their ploidy, that means increasing their chromosome number, or by becoming binucleated, having 2 nuclei. This lets them scale up their metabolic machinery to handle greater demands without the risk of entering an unnecessary division cycle.
The coordination required here is just. It's mind-boggling. An organism might increase its overall volume by a factor of 10? That's a billion-fold.
From a fertilized egg to an adult, yeah.
So if that's happening, how do all the individual parts, the limbs, the organs, the head, stay in perfect proportion through that massive scaling process?
This is the problem of coordinating parts. Scientifically, it's known as allometry.
Allometry.
And it fundamentally suggests that there have to be mechanisms that are capable of sensing the organism's overall size to, modulate the growth rate of individual components.
Okay.
As developmental biologists, we often use a power law equation to represent this data. It's usually written as y equals x to the power of b.
And when I first see an equation like that, it looks like some fundamental physical law, like it's governing biological proportions.
And that's exactly the conceptual trap we need to avoid.
Oh, okay.
That equation, y equals x to the b, is purely a mathematical convenience. It allows researchers to plot and analyze correlations in growth data, but it does not explain the underlying biological control mechanism.
So it's descriptive, not explanatory.
Precisely. If it were a fixed mathematical law, growth would be simple, but the biology is far more complex.
Can you give us a concrete example of how biology must be doing the actual controlling instead of just following a mathematical script?
Absolutely. Let's look at the fruit fly Drosophila. We can look at two separate body parts.
Okay.
The size of the fly's mouth palps, for instance, is highly proportional to its overall body size. If the fly is large, the palps are large. They fit that allometric relationship where B is close to 1.
A simple direct correlation.
Right. But then look at the fly's genitalia. In a large fly, the genitalia are proportionally smaller relative to the body than they are in a small fly.
So the relationship is inverse in a way.
Right. It shows that the regulatory rules governing the genitalia must actively suppress growth or at least scale it down as the total body size increases. This proves that growth isn't just proportional math. It requires a complex biological mechanism that's continuously sensing both nutrient resources and the relative size of its neighbors to achieve that final precise shape.
So if the body needs this sensitive mechanism to sense resources and initiate growth, what are the primary molecular controls? What dictates whether a cell should grow, divide, or just increase its mass?
Well, our sources point straight to the insulin and TOR pathways as the master growth switches. They're like the resource budgeter for the cell.
The central command?
They really are the central molecular nexus. They link the organism's metabolic state, so its nutrient availability, directly to cellular growth and proliferation.
And these are ancient pathways.
Incredibly ancient. The insulin pathway and its downstream target of rapamycin or TOR pathway are found in everything from yeast to humans. That just underscores their fundamental importance.
Okay, let's break down the signal cascade step by step. Where does it start? What's the initial trigger?
The signal begins when insulin, or the closely related insulin-like growth factors, IGF-1 or IGF-2, bind to their specific receptors.
On the cell surface.
On the cell surface. These receptors, like the IGF-1 receptor, are large transmembrane proteins. They span the cell membrane and have these internal tyrosine kinase domains.
And that binding event kicks everything off.
It does. It triggers the activation of the receptor's internal enzymatic activity, which then initiates two major transduction pathways at the same time.
One of those paths leads to cell division. That's the ERK pathway.
That's the promitotic branch, yes. The activated receptor uses specific adapter proteins molecules with names like Shy-C, GRB2, and SOS to couple to a small protein called RAS.
RAS is a famous one.
Very famous, often mutated in cancer. Active RAS recruits and activates another kinase called RAF, which sets off a chain reaction leading to MEK and then finally to active ERK.
And ERK is the final messenger.
Right. Active ERK, which stands for mitogen-activated protein kinase, then moves into the nucleus. There, it collaborates with other transcription factors to turn on genes that are essential for the cell cycle, ultimately stimulating cell division. That's the pathway that increases cell number.
But the other pathway is arguably more crucial for increasing cell size and overall mass. The PI3K pathway. How does that signal start?
The PI3K pathway starts with something called the insulin receptor substrate, or IRS. It gets phosphorylated by the activated receptor. This, in turn, activates an enzyme called PI3K.
Phosphoinositide 3 kinase.
That's the one. And this is where the membrane lipids come into play. PI3K acts on a lipid in the inner leaflet of the cell membrane called PIP2. PI3K phosphorylates PIP2, creating a new molecule called PIP3.
So PIP3 is essentially the molecular signal on the membrane that's screaming, resources are abundant, growth must commence.
Exactly. The accumulation of PIP3 drives the next critical step. PIP3 directly activates another key player, protein kinase B, which is often just called PKB or ACT. PKB is a major positive regulator of growth.
Before we get to what PKB does, we have to acknowledge the critical molecular break on this system, the tumor suppressor, PTN.
This is a vital conceptual point, absolutely. The level of PIP3 in the membrane isn't just determined by PI3K making it.
It's also being removed.
Continuously. It's degraded by the phosphatase PTN. PTN specifically removes that phosphate group from PIP3, converting it right back to PIP2. PTN acts like a dedicated emergency brake on growth.
And the clinical relevance of this is just enormous, isn't it?
It cannot be overstated. When you lose the function of the PTN gene, that brake is cut. PIK3 levels just accumulate uncontrollably,
Leading to chronic activation of PKB.
which drives massive sustained overgrowth and proliferation. This loss of function is strongly associated with a vast array of human cancers, and also with severe overgrowth syndromes like Proteus syndrome.
The condition famously linked to Joseph Merrick, the Elephant Man.
Exactly, though that link is a bit controversial. But the point is, PTN's function as a tumor suppressor makes it one of the most critical checks in this entire system.
That really sets the stakes. Okay, so let's assume the brake is off and the engine is running. We have active PKB. How does this kinase ultimately trigger the central growth determinant, TOR?
This is an elegant system of what we call a double negative, or inhibition of an inhibitor.
Yep.
Active PKB starts this by phosphorylating and inactivating a protein complex called TSC12. TSC stands for tuberous sclerosis genes 1 and 2.
And the job of that complex?
The job of the TSC12 complex is normally to inhibit TO.
Ah, I see. So by inactivating the inhibitor, TSC12.
PKB releases the brake on TR, leading to its massive activation.
So TOR is the powerhouse, but it's always being held back by TSC12 unless PKB gets the signal from insulin.
That is the regulatory architecture in a nutshell. And TR, or the target of rapamycin, is an intracellular kinase. And it's the central growth determinant because it basically controls the cell's entire protein synthesis capacity.
And once it's active, it has two main jobs.
Two main growth-promoting functions, yes. Let's start with the first one, S6K.
S6K.
TOR activates S6 kinase, or S6K. S6K, in turn, phosphorylates a ribosomal protein called S6.
And when S6 is phosphorylated,
It dramatically increases the efficiency and the capacity of the cell's ribosomes, the protein-making factories, to synthesize proteins. This massive increase in factory output is the physical molecular determinant of cell size.
wow. And the second function involves initiation factors?
It does. TOR also inhibits a protein called 4-EBP.
Okay.
4-EBP is normally an inhibitor of a protein synthesis initiation factor called EF1A. So again, you have the double negative,
So stops the inhibitor, 4-EBP.
which allows the initiation factor, EF1A, to start the massive process of protein synthesis more efficiently. So TOR is coordinating both the machinery and the start signal.
That is an incredible level of detailed control. Does this resource pathway also affect the long-term decision to grow at the level of the genome?
Absolutely. PKB has another major target, a transcription factor called FOXO. FOXO's normal role is an anti-growth pro-survival program. It moves into the nucleus and promotes genes that actively reduce protein synthesis. When the insulin-TOR pathway is highly active,
When there are plenty of nutrients.
right, PKB phosphorylates and inactivates FOXO, trapping it in the cytoplasm and silencing that entire anti-growth program.
And if nutrients drop?
If nutrient levels drop and PKB activity falls, FOXO is released. It moves back to the nucleus and actively reduces cell size and promotes survival. It's a beautifully sensitive feedback loop.
So that explains the local cellular control. Now let's connect this to the systemic control across the entire body in mammals, the growth hormone and IGF axis.
Right. On a systemic level, the major drivers of mammalian growth aren't classical insulin, but actually IGF-1 and IGF-2.
Yeah.
They operate through the IGF-1 receptor and feed right into the PI3K-core pathway we just discussed.
And they have different roles.
They do. IGF-2 is absolutely crucial during fetal life, especially for scaling up the placenta, which has to match the metabolic demands of the fetus. IGF-1, however, is the main driver of postnatal growth and size maintenance.
And what determines the levels of IGF-1 circulating throughout the body?
That is primarily controlled by growth hormone, or GH, which is secreted by the anterior pituitary gland in the brain.
So the brain is in control.
It is. The GH-IGF axis is a tightly regulated endocrine loop. A lack of sufficient GH, or a defect in the GH receptor itself, which causes a condition called Laron syndrome, leads to proportionate dwarfism because that systemic signal to grow is just absent.
And the opposite is also true with very dramatic results.
Yes. Excess GH during the developmental period, when the growth plates and the long bones are still active, leads to gigantism.
And if it happens later in life?
If this excess secretion occurs after the skeleton is mature in adulthood, it leads to acromegaly. That's a condition where the bones of the extremities, like the jaw, hands, and feet, enlarge disproportionately. It really demonstrates that even in the adult, these systemic growth signals remain incredibly powerful.
The power of this system is maybe best seen when it's completely knocked out.
Genetic knockouts in mice give us irrefutable evidence. If you delete the GF1 or AGF2 gene individually, the animal's birth size is significantly reduced down to about 60% of normal.
But if you knock out both?
If you create a double knockout, deleting both EGF1 and EGF2, the size plummets to a mere 30% of normal and the organism dies shortly after birth.
Wow.
Similarly, knocking out the IGF-1 receptor or two of the three PKB genes results in an animal about half the normal size, often with perinatal lethality. This entire molecular framework really is the master-scale architect of vertebrate life.
Okay, so the insulin-TOR system handles the metabolic drive and dictates cell size based on resources. It's the accelerator.
That's a great way to think about it.
But organs don't just inflate indefinitely, even if nutrients are abundant. There must be a separate system that's monitoring physical boundaries and density.
There is.
And that brings us to the second major architectural system, the Hippo pathway.
The Hippo pathway is often called the ultimate organ size controller. While 2R manages the individual cell's volume, Hippo is the system that explicitly limits the number of cells based on density and position.
And it was discovered in flies.
Discovered in Drosophila, yes, but it is highly conserved across animals. And just like the PTHrP axis, its loss of function is critically linked to human cancers, which confirms its role as a master tumor suppressor pathway.
Okay, let's detail the inhibitory cascade here.
Yep.
Because it sounds like it shares that same double negative complexity we saw with TOR. It sounds like a cellular committee designed to just say stop.
It is a profound inhibitory cascade. We start upstream with an atypical cadherin called FAT. It's a massive transmembrane protein.
And that's the sensor.
FAT is essentially the sensor on the cell surface. When FAT is activated, often through cell-to-cell contact and density cues, it facilitates the activation of the core hippokinase cascade.
And what are the components of that cascade?
The kinases include the Hippo homolog itself and the Warts homolog. In mammals, these are MS12 and LATS12.
So they're the enforcers.
They are. Once they are active, this cascade phosphorylates and inactivates the central transcriptional coactivator, a protein called YORKI or YIKI.
Which in mammals is YAP or TAZ.
YAP and TAZ, that's right.
Okay, let's trace the logic. If cells are too crowded, FAT is activated. FAT activates the Hippo-Warts kinase cascade. What's the ultimate consequence of this high signaling?
High Hippo signaling means YORKI is heavily phosphorylated and then it's sequestered, it's trapped in the cytoplasm.
It can't get into the nucleus.
It can't. And because YORKI is trapped outside the nucleus, it cannot bind to its partner transcription factors, like one called scallops, to promote cell division and growth genes. This is the stop growing signal.
And if the pathway fails,
then the system breaks down and growth becomes uncontrolled. If Hippo signaling is low, say, if you lose the function of the upstream inhibitor FAT, or if you mutate the Hippo or Warts kinases themselves,
YORKI is free.
YORKI is released, it translocates freely into the nucleus, binds its partners, and drives massive disproportionate organ overgrowth. It's exactly what we see in cancer. The fly wing discs become huge, the eye size explodes. It's a dramatic visual of the pathway's power.
What's truly sophisticated here is how the Hippo pathway links the physical arrangement of cells to growth control through fat. It's not just checking density, is it? It's checking the pattern of the cells.
This is where the physics really meets the genetics. Fat is regulated by two other proteins, DOX2 and 4-jointed. These and FJ.
Okay.
These proteins establish a gradient across the field of cells, giving each cell a specific positional identity. Metaphorically, a zip code.
A zip code, I like that.
And the critical finding is that the activation of fat does not depend on the absolute amount of Ds or FJ in the cell, but on the difference of content between neighboring cells.
So neighboring cells are constantly checking their zip codes against each other.
Precisely. If two neighboring cells have a large disparity in their DSFJ content, meaning there's a major pattern flaw or a discontinuity, it suppresses the full activation of fat.
Which leads to a local drop in Hippo signaling.
Which means YORKI is released into the nucleus, and growth is stimulated in that exact location to correct the map. It's an incredibly elegant mechanism to ensure that the pattern of cells stays coordinated and that any deviation triggers compensatory growth to fill the gap or fix the flaw.
We've covered the global resource system with TOR and the spatial system with Hippo. But researchers long hypothesized that local feedback might exist to fine-tune the proportions. This led to the theoretical concept of chelons.
Chelons, yes. They were proposed as a theoretical class of growth inhibitors. The model suggests that a body part produces a specific growth inhibitor, the chelone, which is then released into the bloodstream and diluted in the total blood volume.
The logic seems simple.
It is. If the organ is too large relative to the body, the chelone concentration is high, slowing its growth. If the organ is too small, the inhibitor level is low, and growth accelerates until balance is restored. It's a perfect, proportional, negative feedback loop.
Did we ever actually find a real example of a chelone that fits this model?
We did, and it's myostatin,
Myostatin.
or GDF8, which is a member of the TGF-beta superfamily. Myostatin is secreted by developing muscle cells, and its function is almost purely inhibitory. It actively reduces the division of myoblasts, the muscle precursors, and it inhibits the enlargement of mature myofibers.
And the consequence of lacking myostatin is visually staggering.
It's the most dramatic real-world example of a chelone at work. The myostatin knockout mouse has a 2 to 3-fold increase in muscle mass compared to a normal mouse.
And that's both more muscle fibers and bigger fibers.
Both. It's a combination of hyperplasia, more fibers, and hypertrophy, larger fibers, and we even see this naturally selected in domestic animals.
Like the Belgian blue cattle.
The famous Belgian blue cattle, known for their immense double-muscled physique. They carry hypomorphic, or less active, alleles of the myostatin gene. It's perfect confirmation that local negative feedback is a very real and very powerful determinant of final size.
Finally, let's pivot to the liver, which seems to have evolved the unique capacity that allows it to skirt some of these rules of finite growth control.
The liver is exceptional among mammalian organs in its regenerative capacity. If a large percentage of liver mass, say 70%, is surgically removed, the remaining tissue doesn't regrow the missing lobes structurally.
Right, it doesn't rebuild the shape.
No, but the remaining hepatocytes quickly proliferate to restore the original total volume.
So this is hyperplasia, an increase in cell number.
Exactly, not true regeneration of anatomical structure. Proliferation begins within 24 hours, and the volume is often restored within just a few days.
How does it know precisely when to stop? It must have a mechanism to titrate its size against the rest of the body.
Early experiments, parabiosis experiments, where you connect the circulatory systems of two animals, showed that circulating factors are key.
How so?
If you partially remove the liver from one partner, the intact liver of the second partner also grows slightly, suggesting a blood-borne signal.
A chelone.
Well, here's the twist. The exact factors identified, like certain bile acids from the damaged liver, often act as activators of proliferation, rather than the inhibitory chelones that were hypothesized. The mechanism might involve activating proliferation when factors drop below a certain threshold, effectively signaling that the functional capacity of the liver is too small.
We circle back to the Hippo pathway, playing a huge role in this unique liver plasticity. It really demonstrates the liver's ability to change its scale.
Precisely. The Hippo pathway regulates this rapid cell number adjustment. If researchers experimentally overexpress YAP1, the mammalian Yorkie homolog, in a mouse liver,
It happens.
the organ can increase its size by a staggering factor of 4 within a few weeks.
4 times the normal size.
But the true marvel is that when you turn the YAP1 transgene off, the liver gradually, but completely, shrinks back down to its normal, genetically determined size.
That's incredible.
No other adult mammalian organ demonstrates this extreme level of controlled cell number adjustment and subsequent scaling, and it's thought to have evolved because the liver is the primary recipient of blood from the intestine. It's constantly exposed to toxins that necessitate rapid compensatory cell renewal and damage repair.
So part 1 has painted a clear picture. TOR sets the resource budget, Hippo manages the physical boundaries, and the liver evolved a special way to use these controls for rapid volume restoration.
That's a great summary.
Now, let's shift gears and look at the most dramatic form of biological repair, rebuilding complex anatomy from scratch. Regeneration is the ultimate act of developmental biology, a total anatomical reset. Why is this ability so highly variable, even among closely related species?
The variability is profound and, frankly, highly frustrating for researchers. A salamander can regrow an entire complex limb, bone, nerve, muscle, everything.
But a frog can't.
A closely related frog loses this ability entirely after metamorphosis. Many specialists believe that the capacity to regenerate complex anatomical patterns is a pristine biological property, one that's genetically complex and, sadly, easily lost during evolution.
Why would it be lost?
Perhaps because of the selective pressure to avoid cancer in long-lived complex organisms. Successful regeneration fundamentally relies on the persistence of complex pattern specification systems, what we call positional information throughout adult life.
Okay, so let's start with the undisputed masters, the planarian flatworms. The classic party trick is cutting one into many pieces, and each fragment regenerates a whole new worm. What's their cellular secret?
The planarian secret lies entirely in a specialized population of adult stem cells called neoblasts.
Neoblast.
These cells are truly remarkable because they're the only dividing cells found in the planarian's entire body. They're considered pluripotent stem cells, meaning they are capable of producing all 12 to 15 different cell types that constitute the entire worm.
So all regeneration comes from them.
Every tissue repair, every new head, is driven exclusively by these neoblasts. If you kill them with radiation, the worm cannot regenerate at all.
When a planarian is amputated, the new tissue forms a blastema. But before any rebuilding happens, the blastema must decide what to build, a head or a tail. How does it determine this primary axial polarity?
This primary decision is governed by the ancient and highly conserved Wnt beta-catenin signaling pathway.
Once again?
It's everywhere. In planarians, Wnt ligands, the signaling molecules, are preferentially expressed at the posterior end of the worm. This spatial expression gradient establishes a clear molecular compass for the anterior-posterior identity.
So high Wnt means this is the back.
High Wnt means this is the posterior, and low Wnt means this is the anterior.
This sounds like a great candidate for an elegant experiment. What happens if you override that signal?
Researchers used RNA interference, or RNAi, to knock down components of the beta-catenin pathway. Essentially, they silenced the key relay component that interprets the Wnt signal.
And the result?
By abolishing the ability to sense the posterior signal, an amputation surface that normally generates a tail, the posterior end, instead generates a head.
A double-headed worm.
You get a double-headed worm. This simple, powerful experiment proves that the Wnt pathway is the critical molecular determinant of axial polarity, and that the positional information required to define a head or a tail is dynamically stored in the cells, ready to be read or rewritten.
Moving up the evolutionary ladder to arthropods, how do insects like crickets handle limb regeneration? And how do they ensure the rebuilt leg has the correct 3D shape?
Insect leg regeneration, which typically only replaces the missing distal structures, relies entirely on reestablishing the fundamental pattern codes that were used during embryonic development.
So it's a replay of the original program.
It is. This involves reactivating the mechanisms that define the three anatomical axes: proxima distal, from hip to toe,
Anteroposterior, front to back.
and dorsiventral, top to bottom.
We should probably briefly recap the core molecular logic here, drawing from the familiar Drosophila imaginal disc system, since that pattern is conserved.
Absolutely. Think of the leg disc as being divided into compartments. The posterior compartment is defined by the expression of the gene engrailed, and cells in this compartment secrete a morphogen called hedgehog, or sh.
Okay, hedgehog from the back.
This signal diffuses forward into the adjacent anterior tissue. The anterior tissue interprets the sh signal, which induces the expression of two other critical morphogens: dicapentaplegic, or DPP, dorsally, and wingless, or WG, ventrally.
So we have shift from the back, DPP from the top, and WG from the bottom. Where does the actual growth happen?
The region that dictates distal outgrowth, the very tip of the limb, is located where the concentrations of DPP and WG reach their maximum intersection.
Where they overlap the most.
Right. This high point of DPP and WG signaling induces the expression of an EGF receptor ligand called VEIN, and VEIN is the direct mitogen. It's the driver that promotes the outward distal growth of the leg. So regeneration has to recreate this intersection point to grow outward.
This model elegantly explains normal distal regeneration. But the truly conclusive evidence that growth is driven by pattern discontinuity comes from experiments creating supernumerary, or extra limbs.
These experiments are crucial for understanding the positional code. If a surgeon grafts a rotated or axially inverted segment, say a piece of leg that's been rotated 180 degrees onto a host stump,
You create a mismatch.
you create an unnatural discontinuity in the established AP or DV codes at the junction. For example, in an antero-posterior inversion, the engrailed positive posterior compartment is now opposed to anterior tissue in two novel incorrect locations instead of just the one natural interface.
That's fascinating. If the normal posterior-anterior interface is what drives the maximum DPP-WG signal, then two extra interfaces should create two extra growth centers.
Precisely. This positional disparity acts as an instruction to grow. The unnatural discontinuities trigger the reinduction of the DPP-WG maximum in two separate locations along the new junction.
That it initiates new limbs.
It initiates the formation of two supernumerary regenerates. These extra limbs always emerge with inverted polarity relative to the central grafted piece, demonstrating conclusively that the pattern mismatch, the gap or clash in the molecular positional code, is the direct mechanical trigger for initiating new, correctly organized growth.
Moving into vertebrates, the salamander or newt is the champion of complex anatomical regeneration. The process here is fundamentally different from the planarian, since it doesn't rely on a constant pool of pluripotent neoblasts. How does the amphibian rebuild a complex structure like a limb?
The amphibian process is sequential and relies on a dramatic cellular transformation, which is why it is so remarkable. It starts with the rapid migration of epidermal cells over the cut surface, forming a protective wound epithelium. The crucial second step is dedifferentiation. Internal tissues, the bone, the muscle, the connective tissue, they all lose their specialized phenotypes. They revert back into a loose-packed, homogeneous mass of mesenchymal cells.
And this mass of undifferentiated cells is the blastema.
Yes. The blastema is a proliferative center encased by the epidermal jacket. The cells divide intensely, and then they redifferentiate, laying down structures in a highly organized proximal to distal sequence. Cone stage, palate stage, early digit, and finally, the late digit stage.
And the signaling is familiar.
Highly conserved. FGFs are expressed in the overlying epidermal cap, acting as the primary mitogens that drive proliferation, and SHISH is expressed in the posterior margin of the blastema, exactly mirroring its function in embryonic limb development.
The reactivation of Hox genes, the master regulators of body segments, suggests the animal is actively reading a spatial map to determine what it needs to rebuild.
That is exactly what happens. Early in blastema formation, genes like HOXA9 and HOXA13 are re-expressed. As the blastema grows outward, these genes set up specific nested domains of expression, with HOXA13 being expressed most distally.
So the cell is using these Hox genes to figure out its exact location.
It's using the Hox code to ensure that a wrist blastema doesn't accidentally form a shoulder.
This leads to the most critical question about the cellular strategy. When bone and muscle cells dedifferentiate into the blastema, do they become fully pluripotent stem cells, like the planarian neoblasts? Or do they retain a memory of their original identity?
This question was definitively answered through fate mapping experiments, particularly using GFP-labeled mesoderm grafts in axolotls.
And the results?
The results showed that the dedifferentiated cells largely retained an epigenetic memory of their tissue of origin. Muscle blastema cells regenerate muscle. Connective tissue cells regenerate cartilage and connective tissue. They do not cross germ layer boundaries or become pluripotent.
So, amphibian regeneration relies on committed, dedifferentiated cells. They're like specialized tradesmen who temporarily put down their tools rather than a universal pool of construction workers.
That's the perfect analogy. They maintain commitment to their germ layer or tissue type. The blastema is therefore a mixture of committed progenitor cells, not a pool of universal stem cells.
And just as we saw in insects, the regeneration process is still driven by the positional disparity, the law of distal transformation.
The law holds that the stump only regenerates the parts distal to the amputation site. But when researchers create an artificial anatomical disparity through axial inversion experiments, again, grafting a rotated segment onto a stump, they reliably produce the original central regenerate, plus two supernumerary limbs at the points of maximum anatomical mismatch between the host stump and the blastema.
The discontinuity itself is the instruction set.
It is. Furthermore, if you force SH expression into the anterior side of the blastema, you get a double posterior duplication, demonstrating that the underlying embryonic patterning mechanisms are fully conserved and reactivated during adult repair.
We've established that the anatomical pattern is stored as a complex molecular code within the cells. If that code is just a set of molecular instructions, is it possible to externally override or even rewrite that code?
It is.
And this is where the stunning results with retinoic acid, or RA, come in.
The retinoic acid treatments are one of the most powerful demonstrations of biological plasticity we have. RA is a natural metabolite of vitamin A and a potent morphogen. Treating a cone-stage blastema with RA causes a powerful, uniform respecification of the limb, acts in a concentration-dependent manner. It's like throwing a switch on the positional code.
Let's focus on the most dramatic outcome. Proximalization. What does RA do to the definition of where the blastema is located?
RA forces the blastema to respecify itself as if it were amputated at a far more proximal level, closer to the body. This violates the sacred law of distal transformation.
How so?
For instance, a blastema amputated at the wrist level, if treated with the right dose of RA, will not just regenerate a hand.
They'll do more.
It will regenerate the missing forearm, the elbow, the humerus, and then the hand structures, resulting in a complete, serially duplicated arm. It forces the wrist cells to adopt the molecular identity of upper arm or even shoulder cells.
And it's dose-dependent.
Perfectly. Higher doses lead to more proximal structures being regenerated, though excessive doses eventually suppress growth entirely.
So RA is literally erasing the cell's current positional identity and making it believe it's further back up the axis.
Absolutely, and it affects the other axis too. We know RA also causes posteriorization and ventralization of the blastema.
And we see evidence for this in half-limb regeneration experiments.
We do. Normally, if you cut off only the anterior half of a limb, it often fails to regenerate well, while the posterior half succeeds. After RA treatment, this capacity is reversed. The anterior halves regenerate fully, while the posterior halves are suppressed.
Why does that reversal suggest uniform posteriorization?
Well, the interpretation is that RA uniformly makes all the blastema cells adopt a posterior code. Regeneration is driven by the disparity between the stump and the blastema. If the RA-treated blastema is uniformly posterior,
It now has a maximal mismatch with the remaining anterior portion of the host stump.
triggering full regeneration. Conversely, if the blastema is already posterior, it finds no mismatch with the posterior stump, and regeneration is suppressed. It's all about maximizing the pattern discontinuity.
This is a fundamental change in identity, a molecular override. What is the precise molecular basis for RA's powerful recoding ability? Which switch is it throwing?
The core mechanism involves RA binding to specific nuclear receptors, which then act as transcription factors. Researchers identified a uridale-specific form, the retinoic acid receptor delta 2, as the key player.
And they proved it with a clever experiment.
An incredibly clever domain swap experiment to prove that this specific receptor, and not just some generalized effect of RA, was responsible for the positional recoding.
Walk us through that domain swap.
They took the DNA binding domain of the retinoic acid receptor delta 2, the part that binds to the genome, and they fused it to the hormone binding domain of an entirely different receptor, specifically the thyroid hormone receptor. They then introduced this chimeric receptor into the blastema.
And what happened when they treated the limb with thyroid hormone instead of RA?
The chimeric receptor, now activated by thyroid hormone, still initiated the signal cascade that normally requires RA.
And it worked?
Crucially, this treatment caused the same powerful proximalization effect, forcing the wrist to regenerate the whole arm. This experiment proved conclusively that the retinoic acid receptor delta 2 is the critical molecule responsible for transducing the positional signal to cause proximalization. It confirms its role as the molecular switch that resets the positional map.
How does it do that?
It acts by upregulating genes associated with proximal identity, such as PROD1 in mice, literally changing the molecular zip code of the regenerating tissue.
The idea that we can throw a chemical switch and force a mature, specialized cell to behave like a much more proximal structure is just the definition of profound biological plasticity.
As we step back from this deep dive, the story of adult development is one of continuous, complex, and highly sensitive control. We've tackled two immense fields today.
On the one hand, we saw that growth is never uncontrolled expansion. It is tightly governed by conserved regulatory networks operating under different functional requirements.
We have the insulin-TOR pathway coordinating metabolism and cell size, acting as the cell's resource sensor, yes. And then the Hippo pathway functions as the structural regulator, monitoring cell density and controlling overall organ size and boundary integrity. Disruption of either of these master switches immediately leads to overgrowth syndromes and malignancy.
And on the other hand, we explored anatomical repair, revealing two distinct yet highly effective cellular strategies.
Right. Tissue renewal relies either on truly pluripotent adult stem cells, the remarkable neoblasts and planarians, or, in the case of amphibian limbs, on dedifferentiated cells that are masters of remodeling but critically maintain a memory of their original tissue commitment.
Ensuring muscle regenerates muscle and bone regenerates bone.
Exactly. And regardless of the cellular source, the fundamental principle for successful regeneration is conserved. It all depends on locally reestablishing the complex positional information codes.
Using familiar embryonic signals like 1 TPP, WG, and SHISH at the wound site.
The organism detects the anatomical disparity, the gap in the map, and initiates precise, organized growth based on that mismatch.
That inherent reliance on reading a positional map provides our final provocative thought. We've seen how specific chemicals, like retinoic acid, can violate what was thought to be a fundamental biological law, the distal transformation rule, by completely recoding the positional identity of the blastema.
And this highlights that the complex anatomical pattern of an adult organism isn't fixed in stone. It is dynamically stored in the cells as a set of molecular instructions ready to be reread and, astonishingly, rewritten when the correct chemical or mechanical signal is applied. The grand challenge now is moving beyond simply observing this plasticity to understanding precisely which molecular switch gives us the most nuanced and controlled power to turn that regenerative capability on in organisms like us, where it has been evolutionarily suppressed.